Everybody, thank you so much all for being here. So what I'm going to talk to you today is mostly from a piece of work that we've recently published, Jivan, Rosmi and I, in the Journal of Theoretical Biology. And but towards the end of the talk, I'll also add in a little bit of work in progress. So that's always exciting for any speaker to test on an audience. But of course, none of this work could have been possible without our funders. And of course, I'd like to thank Dave here and Tobias for their useful suggestions as well along the way to help our work going. So what are we talking about? I'd like to start with the historical note. Now, plants, of course, the history and the future of this planet, their morphology is vitally important in ecology and so on. And plant morphology is ultimately determined by growth at the cellular level. So by growth, what I mean is simply the plastic deformation of the plant cell structure. And there is a type of growth, the type that we care about is called diffuse growth. And in essence, what that means is that the growth happens everywhere along the cell. So roughly how it goes is that the cytoplasm in the cell exerts this turgor pressure, which then induces a tension in the cell wall, and which then causes the cell wall to undergo what's sometimes known as wall creep. It's sort of viscous deformation, and this irreversible process then constitutes what we call plant cell growth. So this process can actually be quite remarkable in terms of scale. I mean, so if you, I guess the model system in plants is this Arabidopsis, and in the root of this organism, here pictured, what happens is that cells are continually being created at the meristem, at the tip of the root, and as they get pushed along, and as they traverse what's called the elongation zone, they sometimes undergo within a few hours an elongation of about 30-fold in length. Now, mechanical modeling of this system has been going on for decades, and one of the earliest examples is due to Lockout in 1965, and it links the relative elongation rate of the cell, so L being the length along the growth direction. It links that to the cell properties. So in this case, links the relative elongation rate to the excess, essentially the excess pressure, T minus Y, via a mysterious parameter known as extensibility. So a lot of the effort, modeling efforts that have gone into this system in the following decades have been to sort of essentially take apart what exactly this phi is. And our contribution to the puzzle focuses primarily on this one component of the cell wall, which has often been overlooked despite biologists knowing it plays a crucial role. So if you look into the cell wall, you find obviously all sorts of molecules. What we want to focus on is something called cellulose microfibril, CMF. And it is known that it's the CMF molecules, which essentially sort of you can think of as inextensible fibers. They give strength to the cell wall. So they are what make sure that the cell doesn't just sort of bulge out like a balloon under the turgor pressure. And so they give the cell this growth anisotropy in one direction. And it's also known that the CMF are dynamic. They move along, or they get moved along in the cell wall. They change their orientation, etc. And given that what we want is to sort of dig into exactly what this extensibility of the cell is, it'd be amazing, it'd be a miracle if it doesn't involve a major contribution from this reinforcing component, this CMF. So we'd like to model the cell wall as a viscous fluid, and it's possibly a good idea to give an immediate intuition into sort of what that entails. So we know that the cell extends axially somehow without expanding radially, and this will be explained by the model. But if you think of the cell wall as a fluid, then of course, naturally, the flow is, I mean, at least a priori, you can't rule out the flow twisting around the growth axis. So here I'm showing you a visualization of how that might go. So on the left here, you've got a cell at t equals 0. And as the wall extends, if you imagine you've put a sort of die somewhere along the top rim, it's probably going to start twisting around the axis of growth. And here, what's visualized is a sort of left-handed twisting growth. And so when it comes to what we want to come out of the model, well, we want at least the following few variables. We want obviously the cell length as a function of time, but we also want the relative twist between top and bottom plate as a function of time. And of course, also we want the CMF orientation as well, and possibly something else, but we'll get into that. Now, this twisting behavior, which we basically get for free out of the model after just thinking about the cell wall as a fluid, is it good? Is it important? Well, we think it is. So it is well known that even at the cellular level, you do get twisting growth behavior. So here on the left here, this is one of the best looking examples in literature I can find of a single internodal cell exhibiting this kind of, you can see this kind of texture on this outer surface of the cell, sort of looking like as it grows, sort of twisting itself into a kind of like a rope. And on the other hand, at the microscopic level, as I said at the beginning, microscopic morphology ultimately all comes down to cellular level growth. So if we can understand the cell level sort of twisting behavior, can we then, would that give us some kind of clue as to how macroscopic helical morphology comes about? So with that in mind, we can now dig into the model. So we think of the cell as a pressurized vessel. And of course, what we're actually modeling is the cell wall, right? So as far as the cell wall is concerned, the turgor pressure coming from the cell cytoplasm is an external force. And so what we've done is we put in as many external forces as, well, basically as we can without making the model basically untractable. So there are a few other forces involved. There's a external pressure here, Q, which is entirely in the axial direction, possibly due to, let's say, other cells, sort of neighboring cells, let's say. And we also have here a big sigma, which represents a kind of a torque-like force. It's actually torque per unit area, but it acts in the azimuthal direction on the cell wall on the top surface. We're assuming, of course, the bottom surface is fixed. And immediately, without loss of generality, we're going to set Px to be 0, right? So the external normal direction pressure, again, possibly due to neighboring cells, we're going to set that to 0. So effectively, we're going to measure all the pressures with respect to whatever Px happens to be. So internally to the cell wall, what do we have? We have a center surface. We have a center surface of the fluid sheet, effectively. And along the axial direction of the center surface, we have an S coordinate. And so here at the top plate is S equals this a priori unknown L, which is of course just the cell length. And the fluid sheet has a thickness H, and of course the something that comes in is the cell radius, so big R, which is defined as the distance from the cell axis to the center surface of the fluid sheet. And we also have a normal direction, which we define to point inwards from the center surface perpendicularly. So that's the geometry of the system. And now we can dig into the actual the constitutive law for the cell wall. And we're not going to plug it out of thin air. We're going to use a well-established phenomenological law from Erickson 1960, which describes a transversely isotropic fluid. So an important point here to make is that it's most helpful not to think about, so we know we want to model the CMF, but it's most helpful not to think about the CMF as this separate entity and this sort of ground matrix, if you will, as this sort of fluid that flows. They're all the same thing. It's the whole thing is a transversely isotropic fluid in the sense that at any point in physical space, there is, in some sense, a preferred direction, and the fluid has different properties in that direction than other directions, and that direction is what we think of as the direction of the CNF, right? The fiber, the reinforcing fiber. And so what's involved in this stress tensor sigma, of course, you've got your Newtonian terms, then you've got this normalized director field, this essentially just a vector field determining the special direction, if you will, within the material. And this term now in red is you can interpret it as the fibers, the CMF, actively exerting a kind of tension. Now we are going to set mu1 to this mu1 parameter. We're going to set to 0 because biologically it doesn't actually make sense to say that the fibers actively exert any tension here. But we're going to keep this mu1 term in here just for completeness and to preserve the original form of this tensor. But here, this term involves the rate of strain tensor projected onto the fiber direction, right? So that gives us a scalar field that we call zeta. And so what this term really, you can interpret it as the sort of stress contribution coming from the fibers resisting flow in the fiber direction, right? So it's a sort of so mu2 here can be interpreted as a kind of extensional viscosity. In fact, that's what we call it I think in the paper, an extensional viscosity. And finally, this big term here, you can think of as a sort of the fiber stress contribution coming from fibers resisting the shearing flow, right? So you can think of mu3, you can interpret as a kind of a shearing viscosity. So overall, this material, the stress tensor comes from is determined by these parameters. So we've got P and mu naught here. I should probably point out as well, this mu naught is you can think of as the ground matrix viscosity, right? The ground matrix, of the cell wall, but of course then modified by the existence of the CNF. And it turns out that this mu naught we really ought to think of as another variable. So we've already mentioned 3 variables that we want to come out of the model, right? We've got cell length, the relative twist of the cell, and of course the CNF orientation. But for biological reasons, we also need to think of this mu naught as another variable. So this, which I'll go into in a bit more detail later on. So anyway, so of course the CNF, as mentioned earlier, they are dynamic. So we use this again well-established transport law for how the fiber orientation changes over time. So you simply you have your advection and you have a term that kind of you can interpret as a kind of a stretching of the director field, and you have a source term that's kind of a orientation. It gives you the sort of a kind of angle orientation effect. But for details, I recommend you go into Green 2008. This particular transport law. And as I mentioned earlier, this mu naught, the reason we want to think of it as a variable is that it is known that the ground matrix of the cell wall becomes more and more highly viscous, more stiff as time goes on, just due to certain chemical reactions going on in the cell wall. So we want to model that as well, and for this, we're simply taking essentially the most minimal possible evolution equation. It's just a transport equation with a constant rate, right? A constant rate alpha that we call the stiffening rate, and of course alpha we'll just treat as a tuning parameter. So here we have the full system. We have the 3 equations that I just mentioned, and of course the standard mass conservation and momentum conservation at the top there. And you will notice that there's no source term in here, so, in equation 1 in particular. And despite the fact that what the cell needs to do as the cell wall elongates is that the cell needs to continually deposit new material into the wall, right? Otherwise, if you think about it, the wall is just going to get thinner and thinner and thinner. But we know from observation that the cell wall actually maintains its thickness, and the way that's done is because is by the cell depositing new material into the cell wall. And yet there's no source term in equation 1 in the mass conservation, and we take care of the sort of material deposition aspect by a boundary condition that we impose on the inner surface of the cell wall. And effectively, the argument is very simple, that you can tune that boundary term just right, so that effectively H, if you remember, the cell wall thickness remains constant. So anyway, so here's the full model, and we of course need to do the standard analysis. So step one, of course, here everything's written in sort of a coordinate-free form, but of course we want to write it in the most convenient spatial coordinate system possible, and that happens to be the S coordinate along the center surface of the fluid sheet, and theta coordinates going around the cell axis, and the N coordinate mentioned earlier. And so you write everything, so you expand all these equations out in those coordinates, and it helps as well to define a flow velocity relative to the velocity of the center sheet, right? So U was just the flow velocity, and V here, we define as the velocity of the center sheet, right? The center sheet of the fluid, center surface, I should say, of the fluid. And the motion, I should say, of the center surface is just kinematic. I mean, we don't have time to go into it, but we can write down three kinematic equations for the three components of V, right? And it turns out once you've done all the rest, you know, the asymptotic and stuff, you will find that the leading order V is just zero everywhere. So, but in general, it helps to think of to look at the velocity as measured from the center surface. So that's what we're going to call little u. And of course, then you non-dimensionalize everything appropriately, and this is the point where we bring in a small parameter into the system. So of course, there is a length scale separation between the cell wall thickness, which is scaled with H, the initial thickness H0 of the fluid sheet, and then there's that's very much smaller than the initial radius R0 of the cell. And in real life, this small parameter epsilon is about 0.01. Okay. I should also mention that the timescale we choose is determined by the initial ground matrix viscosity M0 and the turbo pressure P, which we just set to equal P0 constant, but also epsilon comes into here. I mean, just so that so we get a reasonable timescale, and the this timescale here is about 100 seconds, all right? And then you, of course, you extract leading order behavior from the system. For example, obviously, anytime you have a d by dn, that actually entails 1 over epsilon, etc. I mean, in comparison to d by ds, right? So, so of course, then the geometry of the system just enables you to set anything d by d theta to 0. And here we make a important assumption as well. We assume that the fiber orientation field has only the s and theta components, right? So it doesn't have an n component. Effectively, you can sort of interpret that as we say that, you know, the fibers point sort of tangentially to lie tangentially to the fluid sheet. So effectively, the both components of A, both remaining components AS and A theta, are both determined just by this one variable, right? So one of them is sine phi, the other one is cos phi, and phi here you can interpret as just the angle of the CMF locally measured from the transverse, right? So if phi is 0 everywhere, that means all the fibers are just transverse, and if phi is pi by 2, that means the fiber is longitudinal, vertical. And finally, here we do a thing. We make a move where we impose certain conditions on the parameters in the system to make sure that first of all, the cell radius remains uniformly constant everywhere for all time. So that's R at the leading order remains 1 for all time. And to do that, it turns out what we need is to make sure that the two of the viscosity-like parameters, mu2 and mu3, are large, but not too large, right? Effectively, they have to be quite fine-tuned. And then H0 here, so this leading order thickness of the cell wall also remains uniformly constant for all time. And as mentioned earlier, the way to make sure that happens is you impose that the cell deposits material onto the cell wall at a very, very specific rate. So these are the steps to go through to get the equations to come out. And just to remind you, the equations we're looking for are for cell length L, cell twist, the fiber angle, which now condenses down to one variable phi, right? And the matrix stiffness variable, which is mu naught. So to get there, right, let's try and get cell length and twist to begin with. So the way to get there, so you look at the momentum conservation equation, grad dot sigma equals 0, and you look at the SS and S theta components, and integrate it out spatially and apply boundary conditions appropriately, and you find that that this SS component, right, sigma SS, integrated over the cell wall thickness over n, the n direction, just gives you P minus Q over 2, where if you recall, P is the turgor pressure and Q is the externally, the axial pressure due to, for example, a neighboring cell. And you can do the same thing with this, the S theta direction. You will find that the S theta component of sigma, again integrated over n, simply equals the externally applied torque, right, big sigma. And the other thing you can do, of course, you can dig into the constitutive law. So writing out exactly what sigma SS is in terms of, if we remember, the several viscosity-like parameters, and of course the CNF orientation field. And what you get is this complicated business, all right? So equate both sides and you get these two equations. Now, they look fairly complicated, but actually ultimately they are just simultaneous equations for two quantities, right? For dUs by ds at the leading order and for du theta by ds, right? So because they're just simultaneous equations, you can solve them, and here are the solutions here. So on the one hand, dus by ds, on the other hand, du theta by ds, they're both just slightly different combinations of various items that either look like forces or look like viscosities. Okay, so what are these items? So we have the familiar P minus Q over 2, modified by a mu1 term. Now, remember, we are going to set mu1 to 0, but in general, this is what T is, and S as well. It's this applied torque big sigma, again modified by mu1. So T and S both look like forces, right? So the one is an effective tension, and the other one is an effective torque. And we also have these three items here. They all kind of look like viscosities, but modified by the CNF, right? So the viscosity-like parameters, mu naught, mu2, mu3, are all in there, but they're coupled to the Director field of the CNF, right? So the AS and A theta components, the only non-zero components of the A field. And so they are what we call effective viscosities. And these 5 items go into the definition of these functions A and B in different combinations, and they give you dus by ds or du theta by ds. But it's still not clear exactly how cell length and twist is going to come out of this. And it just so happens that, as luck would have it, once we look into the fiber angle equation, we actually find that if you remember the variable phi that determines the fiber angle everywhere, that happens to be at the leading order independent of S. So what that means is that this whole thing, this A, is independent of S, right? And so is this thing B. And therefore, these two equations can be integrated over S trivially. So all you have is that US at the top end, S equals the cell length, is just simply A times the cell length. And U theta as well at the top end is just B times L. So that's all very good, but also we know just by definition that US, the fluid flow in the S direction at the top end is just dl by dt. It's the rate of change of the cell length, right? And similarly, the azimuthal flow at the top end is just d by dt of the relative twist, which we're going to call big theta. Well, it's that times the cell radius r, which happens to be 1, right? So that really does it, right? So now you equate AL with dl by dt. So what you get is 1 over L dl by dt equals this thing A here. And that is a generalized lockout equation, right? Because if you remember the original lockout equation, all it does is that it relates 1 over L dl by dt to cell wall material properties. But now we have we're digging much deeper into the cell wall material properties, right? It includes these viscosity-like parameters and the orientation of the CMF. And likewise, on the other side, you have 1 over L d theta by dt just equals this thing B. So you have your two equations for L and theta, but of course the system is not closed until you give me evolution equations for phi and for mu naught, because phi and mu naught both go into the right-hand side here, right? And equations for phi and mu naught just come from the very beginning, the evolution equation for the fiber directed field and the evolution equation for mu naught. You go through all the algebra and you end up in these places. All right, and turns out I needed one that they're both equations of n and t. All right, now in particular, this mu naught equation is so simple that it can actually be solved exactly analytically. I mean, implicitly still, because the solution is given in terms of this unknown L. But that's helpful because that uncouples this equation from the rest of the system. So effectively, what remains to be solved numerically is just the system of 3 equations for L, theta, and phi. All right. So now I think we're ready to see some actual results. So this first picture is a typical evolution of the fiber angle as a function of n, which goes along the sort of across the thickness of the cell wall and of course of time. And so what the way this particular system is set up is that initially the fiber angle, the phi, is 15 degrees everywhere. All right, so at time equals 0. And also the boundary condition, remember the fibers are being continually deposited at the inner surface of the fluid sheet, right? So that's where n equals 1/2. The boundary value is also sort of fixed. So we are imposing phi b, the boundary value, to be exactly the same as the initial value of phi, right? So as time goes on, the vertical axis, what happens, of course, is that the fibers that are initially there, they occupy a thinner and thinner region of the cell wall, right? Because later deposited fibers get pushed, or they push sort of outwards, right, towards the outer surface of the wall. And the fibers that are initially present, they reorient more, just because they've had longer to interact with the fluid. So but within a few hours, they become basically longitudinal, right, 90 degrees. And that's not a coincidence. I mean, you can look at the equation and try to find the steady state, and you'll find there's only one attractive steady state, which involves the phi value at n equals -1/2 being exactly pi by 2. And also this is good, in terms of matching data, because it is observed that as cells grow, CMF become more and more oblique and then towards longitudinal as well. So here's another view of this evolution, right? So we're looking at a couple of snapshots, I guess. So if you just focus on the top row for now, a snapshot early on and a snapshot much later. So if you look at the dashed dot line, that's where the initial fibers are all at 15 degrees. All right. Now, what's interesting is that if the initial fibers had been, say, 5 degrees, then they reorient more slowly, as you can see by comparing the two relevant lines, for example, here, or here. So, if the initial fibers had been 10 or 15 degrees, they pretty much settle into their steady state within a few hours. But it takes much longer if the initial fibers were closest to transverse. Okay, and the other two variables, cell length and cell twist, are down here. So typically what we see is that within a few hours, the cell grows by an order of 10. So that's exactly what's observed in real life. And the relative twist is interesting. So we should spend a bit of time here. So firstly, to enable comparison, I'm actually plotting the twist per unit length, right? So what happens typically is that that quantity sort of stabilizes over time. So that doesn't mean the cell stops twisting, right? It just means that the twist per unit length tends to a constant. So when the cell grows by another, x, it twists by another constant times x, right? So sort of the cell settles into a kind of uniform helix, if you will. And another thing that's interesting is that the fibers, in this case, they sort of remain at positive angles, and that means the fibers themselves form a kind of right-handed helix, right? I invite you all to sort of hold out your hands and visualize with me. So the fibers sort of form a kind of right-handed helix around the axis. And of course, what that does is, the fibers encourage flow perpendicular to them, right? So that encourages the flow to go left-handedly, right? So that's why we end up with this sort of twisting cell that twists left-handedly, right, with a negative value for big theta. And it's worth pointing out, and in this, on this page, what we have is the applied torque, big sigma, is 0. So there's no applied torque trying to twist it, right? Every twisting behavior, it comes only from the internal effect of the CMF, right? And in this case, the system has a nice symmetry, which is reflective about phi I equals 0. And what I mean by that is, if we had done phi I equals, say, minus 15 degrees, right? So the initial fibers are form a left-handed helix at 15 degrees, then it will have no effect at all on the cell length function over time, right? But the twist will be the other way. And so would the CMF orientation, right? So effectively, these three figures will be flipped about the horizontal axis, right? So that in that case, your fibers will remain having a kind of retain a kind of left-handed orientation, and they will keep encouraging the flow to go right-handedly, and you'll end up with a right-handed twist of the cell. Now, as soon as you apply a torque, as soon as you have a non-zero big sigma, that symmetry breaks. So here, everything's the same as before, but we have a small positive sigma, right? So the positive sigma on the top plate sort of is trying to twist the cell right-handedly, right? So if you look at then what happens, for example, let's just focus on this bottom right figure, right? So if the initial fibers are left-handed, so minus 5 degrees, that will encourage the flow to go right-handedly. And of course, so does the externally applied torque. So overall, of course, the cell does this right-handed twist, right? But interestingly, if you had an initial CMF configuration that's sort of 5 degrees, so right-handed, which is sort of, and by the way, the CMF sort of remain right-handed for all time, all right? That's this solid line here. They retain sort of positive angles. So they keep encouraging the flow to go left-handedly, but that acts in the opposite direction to the applied torque, which tries to twist it right-handedly. So overall, those two effects kind of almost cancel out, and you just end up with a cell that pretty much grows without twisting. Right. So this, we think gives you a sort of clue into how. Sorry, just motorbike. It gives you a clue into how the organism might coordinate sort of cellular growth in terms of twisting one way or another, or no twist at all, right? Okay, so the results so far I've shown you all involve fixing a kind of a boundary condition where the angle of fiber deposition is fixed over time, all right? But it is well documented again in literature that that's not always the case. I mean, in fact, most of the time, the angle of new CMF fibers being deposited is a function of time. So it starts off when the cell starts to grow, new fibers are deposited close to transverse, right? So if fibers are transverse, they enable fast growing, essentially, right? And later on, when the cells sort of towards the end of its growth, the fibers that are being deposited into the wall become more and more oblique or even longitudinal. That's the observation. And here, what I want to emphasize really is not really so much the results, but the fact that the system is set up in such a way that you can put any phi b into the model. So if you tell me, okay, well, actually, I think phi b is a sort of a smooth step function of time, where it starts off being 0 and goes towards 90 degrees. I just put that into the system, and I can see what comes out. What comes out is that, firstly, growth is in general slower. I mean, that's just because more of the fibers through the wall are sort of more oblique or longitudinal than it otherwise would have been. But and also other observations about how the fiber angle sort of changes over time, etc., that you can make. But the real point here is that you can test any kind of phi b you'd like. Okay. So I think at the beginning, I think probably in the abstract, I promised what's called a generalized lockout equation, and we did derive it, right? So if you remember, the 1 over L dr by dt is this complicated thing involving all the effective forces and the effective viscosities. Now, all of that assumes that you have a constant applied torque, this big sigma that's applied on the top plate, right? But actually, if we allow the cell to sort of, coordinate its growth, or if we allow the organism to coordinate the cell growth, why not? Sorry, we just slipped a slide. Why not allow the sigma, the applied torque, to be a function of time, right? So in fact, you can fine tune the big sigma, the applied torque, can be fine tuned so that d big theta by dt is always 0. So that's the relative twist. So it can be so the applied torque can be fine tuned so that the cell just grows straight. It doesn't twist around its axis. And when that happens, this generalized lockout equation simplifies down to this. All right, so delta P is P minus Q over 2. That's your excess pressure. And so if you want to fit this into the original lockout paradigm, you just conclude that actually wall extensibility is just this 1 over this combination of viscosities and CMF orientation. And this is nice as well, because if you just set phi, the fiber angle, to 0, then AS becomes 0, and you recover 1 over 4 mu naught, which is a previously established result by Dyson Jensen 2010, right? I think. So that's nice. That's a good sanity check. But in general, I see this is this is what the wall extensibility would be in this model. And we can go one step further as well. So what if you allow sigma to be fine-tuned so this big theta remains 0? Why stop there? You can also, if you don't want if you don't want big theta to be 0, but you want theta to be a constant multiple of L. So essentially, you want the cell to twist into a uniform helix, right? So not like a helix that has a bigger step size near the bottom and a smaller step size near the top, but constant step size everywhere, right? And that step size you capture in some prescribed constant lambda. You can do that, right? I mean, that's the fine tuning of sigma that's needed. And then, if you go through all the algebra again, what you discover is that the generalized lockout equation now reads that, right? So it's still this excess pressure, and now the wall extensibility involves this lambda parameter as well. All right. So this, we think, sort of clues you into how sort of the organism might coordinate a cell to grow into any kind of helical shape it wants, either just theta equals 0 or some constant sort of helical step size. And in which case, you can still fit the whole thing into the original lockout paradigm, where the relative elongation rate is proportional to the excess pressure, with a proportionality that in this case involves all the bells and whistles, everything, so the viscosity parameters, the CMF orientation, and whatever lambda you impose. All right. So just to finish up, here's just an example of visualization of what this lambda does. So here are just 4 cells, all with the same initial condition and snapped at a particular time, at the same time. So here we have a cell where the cell is trying to grow into a shape involving lambda equals a small positive number. So that's a right-handed twist. And this cell next to it is just trying to grow into the same amount of twist, but the opposite way, left-handedness. And I should say that everything else is exactly the same between these 2 cells, but one of them grows a little bit more within the same time than the other one, right? That's because the initial fibers are right-handed, so that encourages left-handed growth. And that's why the left-handed growing one grows faster. In fact, this effect is more strongly felt here. So between these 2, between 3 and 4, everything's the same, except one of them has lambda is 0.15, the other one has lambda being minus 0.15, right? Everything else is the same. So because the fibers are initially set up to be sort of right-handed, they encourage left-handed growth, and that's why this cell 4 grows faster. So anyway, so those are all the results I've prepared in the slides, and so I'm not quite sure how I'm doing with time, but thank you all for listening, and I do want to welcome all questions. Thank you.